elements of how this process takes place. And this is the thalamus. Throughout the day, you are constantly being bombarded by sensory information. And there would be no way to make sense of it all if you were unable to streamline which elements of the sensory information was processed at particular times. For instance, in this picture, I can tell you to focus on this watchtower. Now, if you're focused on this, the likelihood that you really can tell the details of this M that I just circled are very unlikely, even though that information is entering your retina. And so the reason that I highlight this is because the thalamus is important in looking at some elements of how this process takes place. And this is the thalamus outlined here. Because it's the site where all sensory information except for smell is processed before getting to the cortex, it helps to organize and filter information that is then processed by the cortex. It is also involved in regulating sleep-wake states. Now, as a relay station for a massive amount of information, it isn't surprising that it is split into many subdivisions. So we're going to outline some of the important ones here. Sensory information from the body enters into the ventral posterior lateral nucleus before going on to the somatosensory cortex. Likewise, sensory information from your face enters the ventral posterior medial nucleus before also going right back to the somatosensory cortex. Now, one way that you could easily remember this association is that for the VPM, you put makeup on your face, and then by default you will remember that the VPL has to be for the body. And maybe you can even think like VPL, the L stands for legs. So that's just an easy association that you can make. Now, vision, this is my beautiful drawing of an eye, enters into the lateral geniculate body before projecting to the primary visual cortex, which is in the occipital lobe. And hearing, this is my ear, enters through the medial geniculate body through a very complicated pathway that involves the vestibulocochlear nerve, and then projects to the primary auditory cortex, which is deep in the temporal lobe. Now, a good way to remember this one is that the L in lateral geniculate stands for light, right? So that's vision, and then the M in medial geniculate stands for music or hearing. It's time for a flash quiz, everyone. Now, how does leptin act on the hypothalamus, and what behavioral effect does this cause? Leptin is a hormone secreted by adipose cells which inhibits the lateral area and activates the ventromedial area. Its activity in the hypothalamus leads to feelings of satiety. Basically, you feel full. Now, let's talk about the limbic system. This is probably one of my favorite systems in the brain. It refers to the connection between various areas of the brain that mediates one's emotions. This includes the cingulate gyrus, which we see there, the amygdala. This includes the cingulate gyrus, which we see right here, the amygdala. You can get also the hippocampus, mammillary bodies, fornix. All of these areas are important in the limbic system. Now, you can think of the limbic system as being responsible for driving all motivational behavior. If you think about anything you have ever wanted to do, right? Have sex with that girl or boy, eat, whatever it is, that feeling that drove you to or away from something was generated by the system. Now, one way to remember the functions of the limbic system is through what's known as the famous 5 F's. Those are feeding, fleeing, fighting, feeling, and since I was told to keep my lectures PG, we will substitute sex for the last F word. Pun intended. The cerebellum, shown in this MRI, means little brain in Latin. The cerebellum integrates sensory inputs from the brain and spinal cord and uses it to coordinate smooth motor movements. It is important to know that people with disorders in the cerebellum have problems with which two things? Balance and coordination. So let's take a look at a patient.
David, I want you to lift this leg as high as you can and bring that heel right on the kneecap. Right on the kneecap. And run it down the shin. Okay, can you do that one more time? High up, right on the kneecap. And run it down the shin bone. Okay, and I want you to use your heel like that and run it back up. Okay, just go down for me. And back up on the shin bone. Okay, good.
Now you'll note that in the video, the patient has a lack of smooth linear movement. Another way this is often tested in the hospital is with the finger-to-nose test. Again, due to the cerebellum's function in coordinating movement, it is difficult for patients to touch the doctor's finger and then touch their own nose. Here we are looking down at the superior surface of the cerebellum. It's almost as if you had cut away the entire cerebral cortex. You can see the tectal nuclei of the midbrain here, but we'll talk about that a little bit later. Now the cerebellum's anatomic divisions include the midline vermis. This mediates truncal coordination, the intermediate parts that mediate limb coordination, and the lateral parts that mediate voluntary motor planning of the extremities with the use of sensory information. The output from the cerebellar cortex is carried to the cerebellar nuclei via Purkinje cells. These are really important cell type that you hear about a lot in neuroscience. There are 4 nuclei in the cerebellum. The dentate nuclei receive input from the lateral cerebellar hemispheres. The emboliform and globose nuclei receive input from the intermediate hemispheres, and the fastigial nuclei receive input from the vermis. Fibers from the deep nuclei send information to the cortex via the superior cerebellar peduncle. One way to remember the nuclei is don't eat greasy food. Don't eat greasy food. One important principle is that the cerebellar hemisphere mediates motor movements on the ipsilateral side. This makes it different than the motor cortex, which mediates movements on the contralateral side. Now the reason for this is because the pathways cross twice. They cross once when ascending through the superior cerebellar peduncle, and then they cross again through the pyramidal decussation in the medulla. So they start off on one side and then they end back up on the same side. Thus, if you get a question where the cerebellum is lesioned, the right answer will be the one that shows a deficit in motor coordination on the same side of the lesion. This is in contrast, as I said, to the cortical lesions, which will show deficits on the contralateral side, as that information crosses once through the midline during its journey from the cortex to the periphery. Now, the basal ganglia is a very interesting system with functions that include motor control and habit formation. It can definitely get complicated, but it is worth understanding because it provides real insight for diseases such as Parkinson's. Now, the basal ganglia mediates descending motor systems. Deficits in the basal ganglia produce either hyperkinetic or hypokinetic disorders. It just depends on where the specific lesion is. And so the basal ganglia, as we're referring to it, comprises of a number of different structures. You have the caudate nucleus, you have the putamen, the globus pallidus, you even have the subthalamic nucleus, and then you have the substantia nigra. Now, just to clarify a few terms, the striatum is essentially talking about the combination of the caudate and the putamen. So, if you can look in the background here, when you talk about those two in concert, then you're talking about the striatum. Likewise, when you refer to the lentiform nucleus, you're referring to both the putamen as well as the globus pallidus. So, this combined structure here. There are 2 pathways of the basal ganglia. The direct pathway, which promotes movement, and the indirect pathway, which inhibits movement. The trick to remembering the complex interactions between the pathways is to think back to a basic mathematical principle. A negative times a negative equals a positive, and a positive times a negative equals a negative. Now, with this in mind, let's look at this diagram. We'll go through the pathways and I'll try to outline things so you can see everything in its anatomical context for these circuits. So the striatum, as we discussed, is comprised of what 2 structures? Again, it is comprised of both the putamen as well as the caudate. And it receives excitatory input from both the motor cortex, here, as well as the substantia nigra, here. Now if we zoom in on this part, we see that there are neurons in the striatum with both D1 as well as D2 receptors. D1 receptors are excited by dopamine. Now these green arrows, remember, mean something that's excited, whereas the red arrows mean something that's inhibiting. Dopamine on D2 receptors inhibits those neurons. So when we look at the direct pathway, we see that in the presence of dopamine, these cells in the striatum are activated. Now when they're activated, they inhibit the GPI. Now the GPI normally would inhibit the thalamus. So what we have here is a negative, right? Inhibition times inhibition, which leads to excitation of the thalamus. Let's go through it again. Dopamine acting on D1 receptors leads to excitation of these D1 neurons. When these neurons are excited, they inhibit GPI neurons. Because these neurons are inhibited, they are no longer able to inhibit the thalamus, which means the thalamus can be excited. Okay, so now let's deal with the indirect pathway. Now these neurons have D2 receptors. And these neurons, when they respond to dopamine, are inhibited. So you have a neuron here in the striatum which receives dopamine, which will then inhibit this neuron. Now this neuron normally would inhibit neuron in the GPE, which normally would inhibit the subthalamic nucleus, which excites the GPI. Okay, so let's look at what's happening here. Dopamine is acting here to inhibit this neuron. Because this neuron is inhibited, it can no longer inhibit this neuron. This neuron, because it is disinhibited, inhibits the subthalamic nucleus. The subthalamic nucleus can no longer excite the GPI. Because it's no longer exciting the GPI, which normally would inhibit the thalamus, can no longer perform that function, and the thalamus is excited to excite the motor cortex. So what we see is dopamine acting on both the D1 and the D2 receptors serves to facilitate movement. And it does so by inhibiting the indirect pathway, which normally inhibits movement, and exciting the direct pathway, which excites movement. So like I said, it can be complicated, but just look at it a few times and it will come together. It is helpful to think of the overall effects of each pathway and then work backwards to the details of each connection, remembering that inhibition of an inhibitory neuron leads to activation, just like a negative times a negative equals a positive. Okay, that was a lot, so I think it's time for a flash quiz. A patient presents with difficulty making coordinated movement with her right hand. A lesion is found in the cerebellum. In which hemisphere would it be found? Now this should be a pretty easy one based on what we discussed. Remember that the cerebellar hemisphere mediates motor movements on the ipsilateral side because the pathways cross twice. They cross once through the superior cerebellar peduncle and then again through the pyramidal decussation. This is where the corticospinal tract also decussates in the medulla. Now that we've thought about the role of dopamine signaling in the basal ganglia, let's consider Parkinson's disease. Now, Parkinson is caused by the loss of dopaminergic neurons in the substantia nigra, and it is a bradykinetic movement disorder. For step 1, this is a classic type of pathology picture where you would be expected to look at the midbrain, which is what we're looking at here. This is the midbrain. And you should be able to recognize that there is diminished substantia nigra pigment and that this patient has Parkinson's. For comparison, the midbrain on the left is for someone who does not have Parkinson, and you can see the darkly staining substantia nigra there. And then obviously here you see a lack of staining, which is indicative of Parkinson's disease. So let's watch a video of a patient with Parkinson's disease. Now you should be able to recognize in this video this pill rolling tremor, which is a real hallmark of Parkinson's disease. Here's another video that I want you to take a look at.
Let's see you walk a little bit for me. Okay. Just turn around and walk towards me. Turn around. Okay, turn quickly. And turn quick. And quick turn. Good. Okay. Okay, that's good. Just walk back a little bit. Back, walk towards the dot there.
Same way.
Uh-huh. Just normally, just walk normally. Turn. And come back. And turn. Good.
Okay, so what I hope you were able to gather from that very long video is a few things. The first is that you should be able to notice the patient has a stooped posture. And one really random thing that I do to help me remember this is I picture a bent P. So that's the first thing, the stooped posture. The second thing is the Parkinsonian gait. It is notable for the slow initiation, but once the patient is able to start walking, he can speed up and even find it hard to stop. When patients turn, they also take more steps than others would to complete that turn. You should be very familiar with how these two clinical presentations look, as they will often be given as part of a question that requires you to deduce that the diagnosis is Parkinson. The mnemonic we've provided for Parkinson is TRAP, which stands for tremor, cogwheel rigidity, akinesia, and postural instability. The diagnosis is made clinically. However, the classic finding on autopsy is depigmentation of the substantia nigra. That's what I showed you earlier. But under the microscope, what you might also see are Lewy bodies. You can see those pictured here where the arrows are. Now, do you remember what Lewy bodies are made of? Lewy bodies are composed of alpha-synuclein inclusions. Now, before we move on, let's think about the pathophysiology for a second to see if there's an intuitive way to understand this thing. The major problem in Parkinson is thought to be a lack of dopamine from the substantia nigra. Now, as we discussed, dopamine from the substantia nigra acts on the basal ganglia to facilitate motion, and it does that through activating the direct pathway while inhibiting the indirect pathway. So it makes sense that a lack of dopamine would result in problems initiating movement, since dopamine acting on this system serves to facilitate movement. Huntington's disease is a hyperkinetic movement disorder. It is an autosomal dominant trinucleotide repeat disorder, meaning that offspring will have a 50% chance of inheriting the gene regardless of gender. Symptoms are caused by lesions in the striatum, specifically the caudate nucleus. The neuronal death in the caudate is due to NMDA receptor binding and subsequent glutamate toxicity. Symptoms include chorea, aggression, depression, and dementia. On brain imaging, diminished caudate nuclei leads to dilation of the lateral ventricles. Here we see a normal coronal section of the brain. You will notice the striatum here. Now let's take a look at a coronal section from a patient with Huntington. You can clearly see a difference, I hope. The caudate is greatly reduced, leading to an expansion of the lateral ventricles. When you see a pathology picture like this, if the lateral ventricles look abnormally large, you should definitely be thinking about Huntington. On a genetic level, the problem arises due to CAG repeats. These are inserted into the Huntington gene. With each generation, the number of CAG expands. This causes further destabilization and earlier onset of the disease. This is a process known as what? I left you in anticipation, and that's the name of the process. While we are talking about Huntington's and its trinucleotide repeats, are there any other diseases you can think of that are also caused by trinucleotide repeats? Well, there are quite a few actually. So we have Fragile X, which has CGG repeats, myotonic dystrophy, which has CTG repeats, and Friedreich's ataxia, which has GAA repeats. It is important when you are studying this material to always look for unifying themes and mechanisms. This is the type of thinking that is critical for success on the boards.